Hello and welcome to another episode of the Omics Cast. My name is Albert Vilella, and for those of you that don't follow me on Twitter, here's my Twitter handle, twitter.com/Albert Vilella. I'm also happy to answer any questions you have if you contact me via LinkedIn, and a reminder that this slide deck is available upon request. And today we're going to talk about the deep sequencing of 3 cancer cell lines on 2 sequencing platforms. Specifically in 2 Illumina sequencing machines, in this paper by Nicolas Robine, Kanika Arora, and others. So in the paper, what they essentially do is report the results of sequencing at great sequencing depth, up to 278x of coverage, a series of cancer cell lines that are relatively well understood and for which they can call somatic mutations, and then compare that sequencing, having done it in 2 different machines, the ILLUMINA HiseqX and the NovaSeq instruments. In general, they say they're consistent, and the 2 machines, in the context of all the available next-generation sequencing instruments that are out there, they're quite similar technologies. So it's not surprising that the results are generally consistent and that there are minor differences at the read level. And then they come up with a high confidence filtering strategy that improves the accuracy of somatic calls. So this is a piece of work that they've done essentially, as many other people have, to be able to calibrate how to do the sequencing on new Illumina machines and extract the highest signal-to-noise ratio from them. And they did this work in the transition from the 2014 released HiseqX machine into a 2017 released NovaSeq machine, NovaSeq 6000 machine, which compared to the HiseqX has a 2-channel chemistry, 2-channel SBS chemistry, where the ATCs and Gs are detected with 2 different channels and a dark base. The G base is a dark base and unlabeled clusters, the G base. And another modification of the NovaSeq at the software level, introduced in comparison to the HiseqX, is that the Q scores, the quality scores of the FASTQ files, are binned more aggressively into 4 quality bins. So what's the difference between 2-channel chemistry and 4-channel chemistry? Well, the difference essentially is due or motivated by wanting to do faster sequencing and cheaper sequencing. If you have to produce 4 different dyes, you have to QC them independently and make sure they're the right quality and make sure they play each other well. If you only have to produce 2 dyes and assay they via 2-channeled lasers of 2 wavelengths, then you have half the work to do, and you can also speed it up by not having to do so many additions of nucleosides through the flow cell. And so the 2-channel chemistry is based on basically 2 channels or 2 dyes, the green and the red channel, and then these clouds of dots here are essentially trying to separate as much the 4 bases as possible. So if this dot here is, this cloud here is maximally separated from the other 3, this one is maximally separated from the other 3, etc., then your data is going to be the cleanest, right? And as you can see here, the A, C, G, and T, the G channel is the dark channel. These 2 here are the ones that receive illumination from one of the wavelengths, and this one receives illumination from both. And so that's what we have in the NovaSeq. The 2-channel chemistry was already tested on a previous platform, the NextSeq platform. And here's an image I took from an early iteration of the NextSeq platform. And as you notice here, the A cloud is closer to the other 3 than in the previous image. So if this A cloud has dots that are close to one of the C cloud dots, somewhere here in the middle, or between the A and T somewhere here in the middle, or between the A and G somewhere here in the middle, then you're going to be miscalling specific bases in some of the cycles because the clouds are not separate enough. And so in the NovaSeq, they introduced new surface chemistry, but also new dyes to try and separate these clouds maximally and get the highest quality possible. And then there's the other aspect of at the software level, the quality scores. Here's a slide that explains the Fred quality score, what it means in terms of probability of something being right or wrong. If you have a Fred quality score of 10, a Q10, it means that the probability of an incorrect base is 1 in 10. So if you call 10 bases, the first one being an A, second one, let's say, being a T, a C, an A, a G, a C, a G, then those 10 bases, one of them, by chance, by the probability of the correctness of your platform, will be incorrect. The other 9 will probably be correct. A Q20, 30, 40, and 50 then increases as such shown in this table. So a Q30, which is the standard people use for quality, is 1 in 1,000, meaning 99.9% base accuracy. Obviously, you want a Q40 or 50 or as much as possible, but most of the machines out there aim at being above Q30. Not too many of them aim at producing close to 40 or above 40. And so this produces a value per base, which when you store it in a FASTQ file, it takes quite a lot of disk space. And so if you have to store many values, the compression rates of that FASTQ file when you compress it is not going to be great, and the FASTQ file is going to be quite big. Whereas if you say, well, anything around a Q score of 2, I'm going to call it 2. Anything around a Q score of 12, I'm going to call it 12. Anything around a Q score of 23, I'm going to call it 23. And anything near 37, I'm going to call it 37. Then you are binning, you're summarizing what you see and making the FASTQ files a lot more compressible. But it also means that if in actual matter your Q score was of, let's say, 32 rather than 37, you're overestimating that base, and vice versa. If your Q score was of 28 rather than 23, you're underestimating it by calling it 23. And so the authors in this paper, they wanted to see if this had an effect on their results. So this is the first important figure in the paper where they show the fraction of mismatches once aligned to a human genome between the two platforms, HiseqX in this bluish color and NovaSeq in this orangish color. As you can see, for many of the transitions, the base mismatches, the results are quite similar between the two platforms. But notably, there is here an area where the HiseqX creates more mismatches than the NovaSeq. So the HiseqX, the 4 channel platform, creates more mismatches than the NovaSeq 2-channel platform. C2T and G2A, remember G being the dark one. And then another one case here where the NovaSeq creates more mismatches than the HiseqX. And here is the T2G, so into the dark one. So different ways of seeing this, but here at the trinucleotide level, the bottom one is adjusted to the difference, who makes most changes. And there's a section here where the NovaSeq does worse. And remember, the NovaSeq is the machine that has the dark G, whereas the HiseqX doesn't have any dark nucleotide. Another important metric is homopolymers, stretches of 20 nucleotides or longer that are of the same of the same base. For example, a poly A homopolymer would be AAAAA, in this case measured 20 or longer. A poly T would be TTTTT, etc. And altogether we can call them poly X's for A, T, C, and G homopolymers. What you can see here in this bluish color is that the HiseqX has more homopolymers for Cs, marginally for Ts, but substantially fewer homopolymers for Gs than the NovaSeq, because the G again is the dark base. And this read 2 on the NovaSeq, read 2 is usually it's usually noisier, but not always, than the read 1, has also more homopolymers in the poly A. Part of this could be simply due to the length of the insert sizes, and this is especially important for applications such as FFP, where the molecules that you're trying to sequence have been degraded by being fixed in formalin. And this poly X plots are also always an important metric to consider when you get your results out of the sequencer, or in this case, the Illumina sequencer. I presume it will be the same for the BGI-MGI technology. So we've seen a little bit what the differences between these two platforms are, but as you can see, people are relatively reassured that they can continue doing their science, and they can take the advantage of scale and speed of the NovaSeq to do their science similarly to what they did before. Reminder, my Twitter handle, twitter.com/Alberto. Please contact me on LinkedIn if you want, and I'll try and answer your questions. And a reminder that this slide deck is available upon request. Thank you very much, and I'll see you in the next episode.